So in recreational health, when we focus on water quality, you might be interested in how do we test it? How do we ensure that the water is safe? So we're going to focus now on how do we test the water where we're not chlorinating. We can't just add chlorine to our lakes or to our oceans because fish and other things live there. We can't just chlorinate everything. So how do we test whether or not the water is safe for people to swim in? So we'll be looking at things in the water that might make people sick. So not all bacteria, not all cyanobacteria, not all protozoa, not all viruses make people sick. In fact, only a few of those will make people sick. So how do we study these to ensure that people swimming in these lakes, rivers, streams, oceans, seas, how do we ensure that they're safe? So we generally use bacteria. So some bacteria require oxygen, they're aerobic. Some don't need oxygen. They don't live out in the lake very long. So they might come from our gut or from the gut of other animals, but they don't live out in the... And then some can do both. We also can measure for the presence or absence of these organisms. Now it's important for us to again remember that not all bacteria are pathogens. So bacteria does not mean disease. Those are two different things. So some bacteria are pathogens, but most bacteria are beneficial. Many of them have some beneficial purpose in the environment. We wouldn't be able to digest our food without them. But some of those same ones that are generally beneficial to us, they're shed through our stool into wastewater, hopefully. And if the wastewater is not treated, we might be able to detect those normal ones that are important for us to digest our food out in the water. If we really want to look for pathogens, we can do that too, but it's like whack-a-mole. How many pathogens are you going to try to measure in the water? We can't measure them all at once. It's really expensive. It's hard. It's not like CSI. CSI isn't real. It's fake. There's a lot of work that goes behind everything you want to measure. So sometimes we might measure broad groups like total coliforms and fecal coliforms historically. If you really want to look for individual organisms, you could. So we'll talk a little bit more about that here in a second. But just introducing you to some scientific nomenclature that might exist out there. If you've ever had any science classes with living things, we write them like this, italics. We don't use typewriters and underlining anymore. The genus name is italicized. The species name is also italicized. This one, though, is capitalized. The genus name is capitalized. And then through papers, you'll see like E. coli spelled out this way. These bacteria will be spelled out, with the shortened genus name up front early. If you had a typewriter, old school, you would see it underlined because they didn't have italics. Or if you had to write it by hand, you can't write in italics, so people would write it this way and then underline it. So you should be able to recognize like which ones are drafted properly too. So as a just understanding kind of the norms in science. So this, what's wrong with it? It's not italicized. This, what's wrong with it? We don't need the underline. This, what's wrong with it? Not italicized. This one, what's wrong with it? Not capitalized. This one, what's wrong with it? Not capitalized. Okay? So generally speaking, we measure E. coli for a reason, and I'm going to explain the reason. So there's these groups of bacteria that live out there in nature, something called the total coliforms, and then there's fecal coliforms that like warmer temperatures. Fecal coliform bacteria live in the gut of warm-blooded animals, and E. coli fit into that group. It's kind of like thinking of like trees. Like there's like maple trees, and and there's like maybe the red maple fits into the group of the maple trees, and then the maple trees fit into the group of the deciduous trees that lose their leaves. So it's just kind of grouping scheme here. So E. coli fit into the big total coliform group. They also fit into the fecal coliform group, and we're not going to spend too much time on the other stuff real quick. Most E. coli are harmless to us. there are a few types that do make people sick, that produce a toxin, but most of them are harmless. E. coli are a type of bacteria. We measure E. coli because E. coli in almost all the coliforms, they exist sometimes in the bacteria, they live in soil, water, and even the gut of animals. But we don't like seeing coliforms in our water. In drinking water, there should be 0. And there's presence-absence testing that can be done. But coliforms, some of the coliform bacteria, they live just fine in the soil or water. They may not have come from an animal. Fecal coliforms, which are not really measured as much anymore because the methods aren't very attractive. Fecal coliforms tend to come from warm-blooded things, warm-blooded animals, often. Again, we don't like to see any of them in drinking water, and you can do presence-absence testing, and there shouldn't be more than 200 in recreational water. But the way that we tend to measure E. coli, or tend to measure recreational water quality for the most part now, is we use E. coli. So there are very few types of E. coli that actually make people sick, but we may treat them like that when we're touching them because we don't want to ingest bacteria we've cultured. We don't want to expose ourselves to them, but they live out in the water because they've been shed there. So the presence of E. coli in the water suggests that fecal bacteria are present. So we don't like seeing E. coli in the water. E. coli should be in the guts of things, not shed out in the water. If it's out in the water, we assume fecal matter, somehow it got out there. So E. coli are mostly limited to warm-blooded animals. They can tolerate living in warm conditions. And again, in drinking water, we don't want to see any. But in swimming water, swimming in the U.S., we tolerate up to 235 colony-forming units. So these little purple dots, these magenta dots, those are colony-forming units of E. coli on an E. coli plate. And then they use Enterococcus, which is a different organism, different group altogether. It's used for marine waters. And then there's companies like IDEX that have these kits that measure the most probable number, the MPN. And it's very colorful. And you can also use this and make sure that it's below 235 most probable number per 100 milliliters. So I'm going to have you watch a video from Vice News on water quality in the Hudson River, and they use Enterococci because you're closer to like a marine body of water. So I'm going to stop this video so you can watch that here in a second, but let's stop.